So we're the Maine Entomological Society. Um, we were formed uh, back in the day at Biles Arboretum. Oh my God, nothing's working. All right, we were founded in 1997, Biles Arboretum in Augusta, and we still have some of those founding members active in the society. Um, we're a society of nature enthusiasts, entomologists, um, birders alike, anybody who likes to go outside and experience nature. Um, so we do research projects. We have a scholarship committee. If if you're interested in that, you can check out our website and find some details on that. That we fund um, coursework. We fund college students, grad students, independent researchers, anybody who wants to do any research on the insect fauna of Maine. We are all for that. And we're for especially for the native and helpful insects of Maine, because pest insects are perhaps at most 50 to 75 species statewide, maybe a few more. Whereas the estimated total fauna of Maine's insects is 20,000 species. But luckily, we have Colleen Teerling here from the Forest Service to tell us a little bit about how we can use some of those beneficial insects to control those relatively few pest insects. So if you're not a member of Maine Entomological Society, but you really enjoy this webinar and want to check us out this summer, become a member, come to some of our events. Our first event is in March. It's maple syruping early season bugging event that's super fun. Come join us. And without further ado, I will hand it over to Colleen Teerling.
Okay. So, before I start, I just wanted to say something about uh, several years ago at one of the the Maine Invasive Species Network meetings. I was at a talk by Nancy Olmsted, who was talking about the language we use when we talk about invasive species and invasive insects, and about being aware of the adversarial language that we sometimes use, and the fact that invader and invasive um, and alien are often used to talk about people and not just insects. And that talk has always stuck with me. And I'm still not entirely sure I've found another word other than invasive species to talk about, insects and other things that are here and don't really belong in our ecosystem. But it's something that I just sort of thought especially now with the way people are being talked about as invaders and invasive and as being our enemies. I just thought I would mention that. So my title was "Insects Are Our Allies," and that is a very sort of adversarial sort of thing. The insects are our allies in the fight against those who don't belong here. Um, there. But you know, some insects can at least help us, but these partially reestablish ecological balance after human beings mess it up. Not exactly a very catchy title. So I still am working on language, but anyway, I just thought I would mention that. This is a slide that I stole pretty much outright from Hillary Peterson, one of her talks, because she illustrates the different methods of biological control. Conservation biological control is probably what most of us do in our gardens. It's good for those especially native pests that tend to attack mass plantings, monocultures in crops and fields, where if you plant things like good pollinator plants or that have good pollen, you might attract parasitoids and predators to your garden, and that might reduce some of the insect problems you may have. And so that is one type of biocontrol. Augmentative biocontrol, that's what I did in a former life when I was working in the greenhouse industry. I was developing biological control agents for use in greenhouses. And if people had a problem with their thrips or with whitefly, they could monitor, and then they could order up a a bunch of sachets of Encarsia, put it out in their greenhouse, and they almost used biological control as a biological pesticide, something that you treat when you need to, and then it may keep things going for a while, but then later you might need to treat again. That works well in really high value crops because it's expensive. So things like greenhouses is often used. In orchards, it's sometimes used. There are some field crops it's used, but not so much. And then out in the wide, wild world of the forest and the fields and the general wild ecosystem, classical biological control is what we use for species that have come here from somewhere else. And if they start creating major problems with human health or the economy, or in most cases the ecology of their new area, they become invasive species. And so then to try to restore some sort of balance, they've left behind all of their natural enemies, diseases, parasitoids, predators. And so to try to restore some sort of balance and kind of tie them in a little bit into their ecosystem so they don't just run amok, um, we will go look in their original environment, look for things that might help um, keep tabs on or keep the population down. And so that's what I'll be talking about in this talk. Um, so almost any time that I talk about biological control, because I do a lot of work with biological control in the Maine Forest Service, um, is if you do introduce yet another foreign species, won't that just make things worse? And to be fair, it's a really valid question, because in the past we have done things that are not smart at all. You talk about biological control in Hawaii, and people will probably boo you out of the room because of things like mongooses were released in order to deal with rats, and then they became a huge problem in and of themselves. In Maine, we've got a lot of things that were not so not not particularly well thought out, because 100, 150 years ago, people were not necessarily thinking about the whole environment. They were thinking, what can I do to stop this particular problem? So the Harmonia lady beetle is one thing that was introduced, and it becomes a bit of a pest in terms of coming into people's houses in the winter. That's not that big a deal if it because it did do a good job of dealing with aphids, um, but what it also did was displace a lot of our native lady beetle and species. And so we have the lost lady beetle poster that's been put out, and yeah, there are lady beetles that we haven't seen for a lot of years, and it's because we brought in another species that was so aggressive that um, it outcompeted some of the native species. And then things like spongy moth, brown tail moth, both that were introduced, one deliberately and one accidentally, similar times. Um, and they're very closely related. People went off to Europe and collected whatever they could that could deal with either or both of those things. Um, and they brought back a lot of diseases and parasitoids, and they were pretty effective against varying degrees of effectiveness, but they did a good job of knocking down the population of spongy moth. So in Maine, spongy moth is not really much of an issue anymore, compared to, say, out in the Midwest, where they're on the leading edge of the spongy moth infestation, and the parasitoids and the diseases haven't caught up, and it's catastrophic there. And for us, it's just it's another insect, and once in a while, when we get drought, things are a bit of there's a problem and a bit of an outbreak, but it's not that big a deal for us. We've got lots of natural enemies for it, but those natural enemies also attack a lot of our native species, like the giant silk moths, the cecropia moths, and the reason we don't see as many of those as you did in the past was because of what we released. And then related to that, also brought over for spongy moth and uh, spongy moth and brown tail moth is the Compsoloria consonata, which is a great little parasitoid, but it's very much a generalist. And both some of the diseases and Compsoloria, they're probably one of the major reasons why we are not just scratching the skin off of our flesh with spongy moth all summer long, every summer, because it does a good job of parasitizing it. Um, but it also has a parasite for a lot of other species of moths, as well as things that are not moths. And one of the things, there's a lot of species of moths that are, some of them very obscure, and probably nobody except maybe some of you Lepidopterus and the MES would even know about them. Some of them more common, but each one had parasitoids that would, that were associated with it. And so you might have 20 different moths with 20 different parasitoids that were keeping them in check. And then um, this comes along, this little tachinid fly comes along, and it's such a good parasitoid that it displaces all of the others. And so now all of our eggs are in this one basket, and it's doing a good job of keeping all of these moths in check. Um, and but if something happens to it, then and all of the native parasitoids are gone, could there be moths that we never even think about that might become problems? And then it's also just a messing up of our ecosystem. So we've done things wrong in the past. We're trying not to do the same thing now. And now biological control does need to go through some really intensive testing. We look at it in its native range of what it attacks and what its host range is. Then we bring it here, put it in quarantine, and we test it against a number of different things with 2-choice tests and no choice tests. Basically, if it's willing to die and not feed on something else, then we can say, okay, it's pretty host specific. It might be safe to release it. But it's a biological system, and nothing's ever 100% certain. So I'm going to talk about winter moth. Biological control in forests usually has a time frame of decades, sometimes 50, 100 years before you get really full, good control. And so it's because the forest is a big place and it's a complex place. Winter moth is a really amazing story because we're finding results from our biological control in a matter of years. So winter moth is from Europe originally, and flies comes out, emerges as adults and flies in the late fall, early winter around Thanksgiving. The male has wings, the female does not. Then the eggs hatch early in the spring, and they're little green inchworms. It has a pretty wide host range. And it also includes some agricultural plants like apples and blueberries and cranberries. So it's an agricultural pest as well as a tree pest. Because it comes out, the caterpillar hatches so early in the spring, it feeds on the leaves often before they're even emerging, before the buds are breaking. And so it can cause a lot of damage. As I said, it feeds early in the spring and then it drops to the ground. And it's in the ground from June right through to November, pupating in the ground. And that's how it came to Maine. We're pretty sure that somebody dug up, probably in Massachusetts, dug up there because they had a big infestation in Massachusetts. Dug up plants from underneath their tree, brought it to their second home, and that's where we started to see infestations in Maine. And it's been a problem in other places. It's been out west in BC, and Ontario, BC, and Oregon and Washington. In the 1950s, it was a big deal in Nova Scotia. 40% mortality in some stands of red oak. Um, and in Massachusetts, killed thousands of trees, tens of thousands of acres of defoliation. So it was a really big deal. Luckily, there is this little fly. There's a little tachinid fly called Sizenus albicans, and it's quite small. What it's sitting on there is not window screen. It's a really fine nylon mesh. So it's a really tiny little parasitoid. And so it was being released in Massachusetts, and then we started as soon as we discovered that we had winter moth. We started to work with Joe Elkington's lab at the University of Massachusetts, and started releasing it here as well. The way this works is in the spring, the winter moth hatches and starts to feed early in the spring. Um, and then the Sizenus comes out, emerges from the soil. It's been pupating in the soil, but it emerges in the spring, and it doesn't lay its eggs directly on the winter moth the way a lot of tachinids do. What it does is it can smell where the winter moth has been feeding, and it lays its eggs on the leaf, on the margins of the leaf that have been chewed, so that as this hungry caterpillar comes along and keeps chewing on the leaves, the eggs get eaten, and the eggs just sit in the gut of the caterpillar. And then when the caterpillar drops to the ground to pupate, that's when the tachinid flies hatch, so that eggs hatch, and then it parasitizes the the winter moth. Um, and so the Sizenus alone only provides about 40, maybe 60% mortality, 40, 60% rates of parasitism, which is not really enough to provide control all on its own. However, remember what I said, that the winter moth spends from June to November down in the soil pupating. There are lots of predators in a healthy soil, things like rove beetles and ground beetles, little insectivores like shrews. And so if you were to have a vast acreage of where people were spraying their lawns to make sure there was not a single grub, not a single insect living in their lawn, and rodenticide, so you had no moles or voles or shrews, you probably wouldn't get really good control of winter moth, because the Sizenus can't control it on its own. All of these generalist predators that are down in the ground, they can't control it on their own either, because they're generalist predators, and a generalist predator can't sort of follow the huge boom of invasive species, and so they just can't keep up. But you put the two of them together, the Sizenus knocks down the winter moth enough so that the ground predators are able to provide the rest of the control, and you can get really good control. It's kind of a cool little system. This is work that came out of Nova Scotia, and then also out of Joe Elkington's labs as well. So how there is no store that you can go to and order up some Sizenus. There's no government agency that's rearing them, no big lab. So Joe was rearing them in his lab down in University of Massachusetts. And then we started to get some from him when we noticed that we had it in Maine. And he would bring, he started out bringing adult flies up, and we were releasing adult flies in big cages. And then we realized that did not work that well. And so we started bringing up pupae in the soil, and then we would release them. And as we started to release them in Maine, we started to develop populations. And so we would go out every spring. We go out. It's all hands on deck for a few days. when the caterpillars are they're finished feeding and they're just about ready to drop. And so because they finished feeding, they've hopefully ingested the Cyzinus eggs. And we go out and with giant tarps, we lay them down on the ground under the trees. We beat the trees with great long sticks and we shake the smaller trees, and the caterpillars rain down, and then we pick them up by the handfuls, put them in buckets, and collect thousands and thousands of them. And then we're really grateful to Joe's lab because they take them down to Massachusetts, and so we rear them up usually, and then they take them down and separate out the parasitized and the non-parasitized pupae and send the parasitized pupae back to us. And we put them in these little cages, bury them in the ground, close them up. We put them in the cages because we want to protect them from all of those insectivores living in the soil that I told you about. And there you can see in the spring, we open up the cages and let them go. In the spring, before we open up the cages, as they're starting to emerge, we put a chunk of apple in there, and that provides them with moisture and some sugar. And we want to keep them in the cage so that they can find each other and mate before we open up the cage and let them out into the great big wide world where they might not find each other. This is a picture of me there rather than Tom Schmelk, who is the lead on this program, because after Charlene retired and before Tom came on, no, it was after. Anyway, before Tom came on, I was doing winter moth in the biocontrol for a couple of years. So imagine Tom there instead of me. So this year then was a little bit different. So in the past, we have gotten, before we had any biological control established, and you say that it's established in Maine, we got all of our parasitoids from Massachusetts. And then Massachusetts, the biocontrol worked so well that you couldn't find any winter moth in Massachusetts. So then we were working on our own and just getting our own biological control. And then their lab helped us with separating out the parasitized and non-parasitized pupae. And then this year, Rhode Island has been having an outbreak, and even though they have released biocontrol in the past, it's not seeming to do a lot, and so they're would like some more biocontrol. So this year we activated the Insect and Disease Compact, which is sort of like our version of the fire compact that wildland firefighters have. Whereas if there's a fire in some state or province, everybody will go and help them fight that fire. And then if there's an emergency in some other province or state, they come here. And so this is a system where the host state or province, the ones that's asking for help, will provide the money for the travel, but the home states and provinces of everybody that comes pays their salary. And so we had a bunch of people come from Vermont and New Hampshire and Rhode Island and helped us do this big push for getting all the biological control, or all of the caterpillar collection that we possibly could. So we had a big group and we collected a lot. I can't remember. I want to say 10, 11 or 12,000 caterpillars. And so it's great because the people that come to help us are giving us some help in collecting, but they're also learning. And as we're all together, we're teaching each other and learning from each other. So that was really good. And I promise not to bore you with lots and lots of data, but I just wanted to put this on here to show you. This is over time. Back in 2013 is when we started this, and we were getting pretty high numbers here. 2, 3, 1 year, I think in 2014, we got over 4,000 Cyzenus from them that we put out, and in later years, slightly lower numbers, but we're still getting pretty good numbers. And then 2019 is when biological control really kicked in Massachusetts, and you couldn't find any winter moth there anymore. And so then we were stuck just using our own, collecting from our own sites, and we just didn't have that many yet. And so we did a lot of collection, but our numbers weren't all that high. And then the numbers started to grow and started to grow and started to grow again. And then this year, even though we collected a lot of caterpillars, and there were a lot of Cyzenus in them, we um, divided them in half and are sending half to Rhode Island, so the numbers are a little bit down. So you can see that the numbers, it's just sort of the numbers that we were working with. And then the percent of the parasitism. And this is what's so amazing. It's really nice that you can just go knock caterpillars out of the tree, and then you can tell what percentage of them are parasitized, and you know just how well things are going. So this past year, this is what we found. In Kittery, the population the population of winter moth is sort of not really knocking down, and we've got fairly low rates of parasitism. We're not entirely sure why, but we released at Kittery, and then we also collect. So we collect at Kittery, but we also collect at Brave Boat Harbor, which is a few miles away. So certainly it has spread. The parasitism rates tend to be, fairly low, usually under 20%. And yeah, we don't entirely know why. South Portland was only 1% this year. In past years, it's been in the 40s. So again, not entirely sure what's happening there. And then some of the sites that we've done releases more recently, especially with some of our small releases. Bath, that was one of the years that I did the release, and somebody vandalized the cage that we set out and opened it up, and all sorts of predators got in. And by the time we realized that and closed it up, there were spiders and ground beetles and everything was in there. So we weren't sure how many we were going to get, and I think I only saw about 7 or 8 adults that emerged. Probably were more than that in the end, but not very many. And still, we're up to 12% parasitism rates already. And then down to South Bristol, where we released in 2023, they're up to 45% already. That's not to say it'll be at 45% every year. South Portland was up in the 40s a couple years ago, and it's dropped down now. So the populations do fluctuate. Something that's really interesting is Mount Desert Island. We've never done a release on Mount Desert Island. It's a long ways away from anywhere we've released, but they've got 19% parasitism. And what we think probably has happened is that, we talk about people that, come to their second homes and bring their hostas from underneath their infested oak tree. Well, they brought winter moth with them, but they probably brought Cyzenus with them as well. So that's kind of cool. So yeah, that's winter moth. That's probably one of the most encouraging biological control stories you've ever heard. And if you want to go home with happy, fuzzy feelings, you should probably just go home right now, because this is the best story I will tell. And none of the others are nearly as exciting and encouraging as this one. There's no silver bullets in biological control, but yeah, Cyzenus comes pretty close. It's fast. It provides really good control. The other thing I should mention is that it's also incredibly species specific. Winter moth has a very closely related native species called the Bruce bandworm. They're so closely related that they can interbreed with each other. But Cyzenus is so specific, and they're so closely related that you can't even tell the difference. The only way you can tell the difference is if you kill them and you dissect them and you dissect the genitalia and look at it under a microscope. So they're really very similar, but still Cyzenus attacks winter moth and not Bruce bandworm. So it's a really good little parasitoid. Hemlock woolly adelgid is not going to be nearly as encouraging a story, but we have been doing biological control for a long time. Um, Hemlock adelgids are just kind of odd um, in and of themselves because they have no parasitoids. They have predators, but no parasitoids. Usually, biocontrol programs involve or rely more on parasitoids because they tend to be more specific. They just tend to be better biological control. But with hemlock woolly adelgid, we have to use predators. And so we have Sasagesgymnus sugae, which was one of the first predators. It's a tiny little beetle in the lady beetle family. You can see it here with its nose into an ovisac feeding on adelgid. And here you can see one of the larvae that is also nose deep in there's a female under there with a bunch of eggs and it's just chowing down. Um, it has multiple generations a year and it feeds throughout the spring and the summer. Um, and it was thought it was going to be a really good biological control. And then it didn't seem to pan out for a lot of states. And most states said, we put it out there, we can never find it. It's not doing any good. Don't waste your money on it. But in Maine, for whatever reason, whether it's because we're coastal, whether it's because we're really stubborn and we just kept looking, I'm not really sure why, but we did find it. It's really hard to find because when you release it, it goes up into the tops of the trees. It never comes down to the ground to pupate. It overwinters up in the tops of the trees. If you have a cherry picker, it's a lot easier to find it than it is when you're down on the ground. But it's hard to find, and so it's really hard to tell what it's doing. But we know we've found it like 18 or 20 years after release. Laricobius species um, is one that has also been released, and we have been releasing. Here you can over here you can see one with its nose stuck into an ovisac. We pulled it out, and so you can see it there. We pull it out and put it on a needle. There are a number of species that matter in of Laricobius. One is we have a native species of Laricobius called Rubidus, and it's an important predator of pine bark adelgid. The first species was released for hemlock woolly adelgid control was from the west coast and was not perfectly speciated yet from Rubidus, and so it does hybridize. And so in Maine, we started to release it, and as soon as we realized it was hybridizing, we said, no. Maine is really tends towards being very conservative about releasing biocontrol. And so we said, yeah, we're not, if it hybridizes, we're not going to release it. And so we didn't. We focused on Sasagesgymnus. And then eventually they started rearing Laricobius osakensis, which was from Japan. And it was far enough, far enough away and far enough speciated from Rubidus that it does not hybridize with any of them because it's from a totally different continent. And so might seem a little bit paradoxical. You might think, oh, well, I really want to release something that's from North America rather than something that's from Asia. But actually, it's probably safer to release something from Asia that is not going to hybridize with our native species. Nathan Havel has done a lot of work on this hybridization, and it looks like the hybridization is stable and at a fairly low level. And so, we're starting to release Nigrinus again in Maine. And then Angela Meck at the University of Maine is doing a really interesting, one of her grad students is doing a really interesting project looking at the hybridization and what does Rubidus do in places where there is hemlock and pine, but no hemlock woolly adelgid. What does it do when there's hemlock and pine, but there's hemlock woolly adelgid, but we have not released any Nigrinus yet? Where is it? Does it go and does it feed on the hemlock woolly adelgid? And then what does it do in places where we have released Nigrinus and there is still Rubidus there? How does it behave? Where is it found? what kind of hybridization is there. So that's going to be really interesting and some really important work to be done. Um, and so we have been releasing biological control. The Laricobius is what we get from the Hemlock Woolly Adelgid Initiative. And so it's U.S. Forest Service and universities, and we only get a certain number. We have to share them amongst all of the different states, and so states and provinces. And so we don't get that many, but Sasagesquimnus can actually be bought. And starting in 2022, landowners along the coast, people who did have a fair bit of money and were able to pay for biological control, started to say, we want to do something. We've had a lot of drought and the trees are declining fast. What can we do? And land trust started to get in on it. We want to do something to protect the hemlock trees and the whole ecosystems that are starting to disappear. And so the numbers that private individuals and entities like parks and land trusts, cities and towns have bought has been really amazing over the past several years. All the more so because there is no guarantee that Sasagesquimnus tsugae will control Hemlock Woolly Adelgid. We don't know that either of those species will control Hemlock Woolly Adelgid. There is some evidence in states to the south of us that both of them seem to provide a degree of control at some sites, but not necessarily every site. But we haven't really gotten to that point in the northern New England states. And so we can't really say, yes, it's going to do something. We've got a colder climate. Is it just that we haven't released enough yet and haven't been releasing them as long as states to the south of us have? Or is it something about our climate, which means that maybe we might not get the same degree of control? It's because it's hard to find Sasagesquimnus tsugae. We could say, yes, it's there, but we only find one or two at a time. A great year, we might find 7 or 8. And we can't really say what it's doing and how many are actually out there. One of the things that is encouraging is that anybody who has worked in Connecticut or Pennsylvania places, they come to Maine and say, why is hemlock so much more catastrophic here than it is in places like Connecticut or even Massachusetts sometimes? And part of that may be because they've been releasing biocontrol for a much longer time. And the biocontrol allows the trees to survive, maybe not thrive, but allows them to survive, whereas up here they tend to be dying. Maybe that's due to biological control. One of the things that's encouraging to me is down on Kittery, where we first started releasing biocontrol in 2004, and the trees there were white with adelgid. Some of those trees are still alive. They're some of the world's ugliest hemlock trees, but they're alive. Whereas in other places, a lot of the hemlocks have died. And also, what's even more encouraging is that young hemlocks are growing up, and there's 10, 12-year-old trees that are growing up. And in general, it's the young trees that die first, and you tend to see no hemlock regeneration when there's adelgid. So, as a scientist, I'm really frustrated because I can't say, if you release these beetles, this is what will happen. But, as a non-scientist, I think, well, maybe that's due to biological control. So kudos to those who've been buying it and releasing biocontrol. Then also we'll mention that silverflies are some sort of an up and coming possibility, and it's being researched. It's still in really early stages, but that's a whole other story yet. And while I'm talking about hemlocks, elongate hemlock scale also knocks down hemlock trees, and between the hemlock woolly adelgid and the scale, they can be really, really knock down hemlock trees and kill them. But we have found this little predator called Cybocephalus niponicus, which moved in on its own. It's a generalist predator. We'd have never released it on our own because it feeds on a wide variety of things, but it was released in other states in the past, and it has moved in on its own, which, if it's going to move on its own, sure, great. And I will not mention that. So emerald ash borer, the biocontrol in emerald ash borer is fairly straightforward. Woodpeckers are a big, important part of biocontrol early on in an infestation, usually the smaller woodpeckers and other birds like nuthatches. Um, and they can cause up to over 90% mortality in individual trees of emerald ash borer, but they can't keep up as the population explodes because, again, they're generalist predators. And so we have three teeny tiny little parasitic wasps, and one of them is Tetrasticus planopenisi. It's a larval endoparasite. It's very small. You can see it up here, and that's my finger. And I've got fairly small hands, so you can see how tiny it is. It's got a very short ovipositor, and so it's best on the upper parts of trees where the bark is thinner or on younger trees. It's an endoparasite. And here you can see all of the larvae or pupae inside the X emerald ash borer. Spathius gallinae is one of the other ones. It's a bigger parasitoid. It's got a longer ovipositor. You can see that there. You can kind of see it here. And it does better with the larger trees because it's got that larger ovipositor. And here you can see the parasitized EAB. And then there's Ubius and grilli, which is an egg parasitoid. And if you see that tiny little speck on the tip of a 1-inch cap, that's the egg parasitoid. It is tiny. We release them by putting them out. Sometimes we release adults. Sometimes we release pupae, either in bolts of wood or what's commonly known as the ubinator for the Ubius egg parasitoids. And then we do parasitoid recovery, looking for, oh, so for the egg parasitoid, you scrape the bark off, put it in little vial or in little rearing tubes to try to collect any parasitoids that might be in there. Incredibly labor intensive, and they're very small, very hard to find. Yellow pan traps is another official way of doing it. We haven't had much luck with that. And then peeling cutting down trees in areas where we've released and peeling the bark off, looking for parasitoids is another way. What we have found works best for us is both the peeling and then parts of the trees we'll peel, parts of the trees we'll put in rearing barrels. And that's where we have had most of our recoveries. And so this is where we have been doing the releases. We've been doing them up north and we've been doing them down south. Down south, we've had most of our recoveries. Up north, found one. We've recovered one Tetrasticus. Up north, it's maybe a little bit trickier to get biocontrol established up there because it was a point source. One spot where EAB was introduced, and it's a 2-year life cycle there. And so the life cycle is synchronized. And 1 year you'll have all the adults come out, and then the next year they'll all be inside the tree and they'll be growing up as larval instars. And then the following year the adults will come out. And so some years for parasitoids, there's lots of prey that are at the right stage for them to parasitoid, lots of hosts. And then other years there'll be very little. So it might be difficult to get biocontrol established until the generations start to get all kind of desynchronized. But we have been finding it down south, and we know in New Hampshire, our neighbors there have found it in most of the places that they've been releasing, and so that's really encouraging. And then we also are doing a research project with APHIS. If we protect mature ash trees with pesticides for a few years as biocontrol established, will the biocontrol be enough to protect the trees all on their own? Because in general, we release the biocontrol on the mature trees. Those trees will die. The next generation of trees that growing up should be protected. And there's evidence in other states that are doing it longer than we have that seems to be working. So this is a project that we've started, and I am running out of time, so I think I will stop there. I was going to talk about a few other species, but yeah. So those are just some of the biological control programs that we're doing in the forest. I was going to talk about some of the agricultural programs and purple loosestrife, but I'm kind of out of time, so I'll stop there and see if anybody has any questions.
All right, yes. If anybody has any questions, you can either unmute yourself and ask, or go ahead and type it into the chat. We had one earlier. I believe it was when you were talking about the winter moth. Um, Barbara was asking, is there any data on Kennebec, Somerset counties for winter moth? Is winter moth in those areas?
Um, there probably isn't some. It tends to be coastal. Um, so I can't. I'm not sure if we've released. It's probably there. We basically can just choose one site each year where we can release biological control, because we just don't have that many, and so we pick the site that's the worst or that is far enough away. We're trying to be strategic and place our biocontrol sites throughout the state so that they can basically spread because we know they will spread. So I don't know if we've released yet in Somerset or Kennebec. I'm pretty sure, in the coastal areas that there probably is winter moth in those areas, but I don't think we've been releasing yet.
Okay. And we also had a question from Kathy Murray. How do you assure that S. sugi is species specific, predating on hemlock adelgid?
Um, so that the work was done long, long ago. It was probably the first biological control we've used for maybe not the first, but one of the very early ones for hemlock woolly adelgid. And so there was work done on it. Let's see, it was the Laricobius, I believe, that feeds on a couple of other species of invasive adelgids as well. Um, but Sisagis gymnosugae just feeds on hemlock woolly adelgid. So that is work that was done in labs long before I'd even heard of hemlock woolly adelgid.
Does anybody else have any questions? Either type it in the chat or unmute yourself.
Hi, Colleen, Joe Staples here. How you doing?
Hi.
Hey. So one of the things that comes to mind is, and you brought this up very clearly in the beginning, is that this is a nuanced approach, right? There are a lot of things that are going after these guys. And of course, we're all rather resistant to using pesticides because they're non-specific, of course. Um, in most cases. And so one of the things that I've seen in the past is sort of masking. And so it's not a pesticide approach per se, although it would have to be treated that way. But I've extracted green leaf volatiles from grass and other things and put it on pine to control pine false webworm, for instance, and seen
Ah.
success with reducing oviposition. And currently we're testing the use of Japanese knotweed extracts, which contain emodin and resveratrol, because if you buy resveratrol, it probably came from Japanese knotweed. It produces a lot. But these have a range of effects on insects, from toxicity to increasing metabolic rates to messing with their pupation and things of that sort. And so I'm wondering if there is a kind of gentler way that we can use local, or I want to, sorry, I'm going to call it knotweed naturalized, but local plants to help sort of mask or deter. So emodin from Japanese knotweed, for instance, deters feeding, excuse me. And I'm really interested in using it to look at emerald ash borer because we had a lot of that down here in southern Maine this year.
That's really
Yeah,
interesting.
yeah, and so you're making me think, combining these multiple approaches because these are not knock me down pesticides. They're just like, don't eat me, you know?
Yeah.
Yeah.
And biological control often may not be, certainly is not a quick answer, and may well work best with other things. And so something like that, yeah, could be a really good complement to
Yeah.
using multiple approaches together. Um,
I, I tend
yeah,
not to see people
get
doing
into
that.
integrated pest management for some of these things, and that's where we're going for as biological control being part of a larger program,
Right.
using all sorts of other things, cultural controls, and yeah,
Yeah. Yeah. Fascinating stuff. Thank you.
yeah, that's really intriguing. Yeah, thank you.
We had another one in the chat from Sven. How can you distinguish a parasitized winter moth pupa from a non-parasitized pupa?
Ah,
Is there a
I
way?
thought somebody might ask that. I don't know the details of that. That is something that Joe Elkington's lab has done for us. Um, Tom Schmelk might know the answer, and I don't know if Tom is on today. If you're in, Tom, jump in. Um, but that's not anything that our lab has ever had to do. They've done it for us. I do know it's a bit laborious. I'm not sure if they're. I don't think it's like candling with eggs or anything like that. But there is a way of basically they have to look at each cocoon and determine whether it's parasitized or not. So it's slow and laborious, and I'm very grateful that they're doing it, but I don't know the answer to that. Sorry. If anybody else here on here does know that, I don't know, Charlene, you don't know that, do you? I know Charlene's worked
When
on winter
I
month.
want to. I'm trying to rack my brain. It's been quite a few years since I worked with it.
Yeah,
Um, so the fly egg is inside, a cocoon is inside the winter moth cocoon, and it may even be that you shake them, or it's a weight thing.
I
Um,
thought, yeah,
I'm not sure.
it's something they can't tell until fairly late in the summer.
Yeah, 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 because the the fly has to make its cocoon
Yeah,
inside
so it
the
has to have
winter moth
already
cocoon.
eaten, basically eaten
Yeah,
the the caterpillar, and it is then in its cocoon, and that then feels different in some way. Whether,
yeah,
yeah,
yeah. So then, Colleen, my question is, like in South Portland, the winter moth population was pretty much under control there, at least in some years. So when you
oh yeah,
see really low um, populations of the parasite, is the defoliation, high again?
it was. So every this was at a private residence, and so every year we get their perspective of it, and they're saying, oh yeah, it's worse this year than it's been in the past, but it still wasn't all that high. Um, so no, I don't think it was all that high. So it has been, yeah, we've had very good parasitism rates there for a lot of years. For whatever reason, it's being lower this year. Um, I do remember one year, and I don't know if it was last year, that birds came through just before we were collecting, and they just cleaned out in that area. Just went through and cleaned out all the winter moth. Did they, collect some for some reason? Were know differentiating and eating more, hunting more of the ones that were parasitized? Unlikely. I don't know. I honestly don't know what the difference is, but it certainly was not catastrophic at South Portland, even though the parasitism rates were so low.
We have another question in the chat from Kathy. So rearing these biological control vectors sounds very labor intensive. It definitely is. How sustainable is it, and does it only happen if you have public funding for it? And if so, is that at risk these days? And I kind of think like what you were telling me before we started the webinar, like this lab in Michigan is basically supplying the entire country and Canadian provinces with EAB parasitoids. So I mean, how do they make that happen? That's such a big process.
One place, yeah. And yeah, so that does put it at risk. Um, for hemlock woolly adelgid, um, we sometimes get a little bit frustrated because Maine is often one of the last places to get some of these insects. And just as we're ready, like we need these things, and then they're on to the next big thing. So it is definitely an issue. And for hemlock woolly adelgid, we've gotten to the point where we can go out to other places further south and collect beetles and bring them back. And that's probably going to be the future for that. Um, I don't know if we're at the point that we could do that with EAB biocontrol. I think if we lost the big EAB lab, it would be pretty catastrophic. Um, and one I didn't get to is purple loosestrife and a project that the Kennebec Area Land Trust is doing, where they're just going down to places where other places where in Maine where biological control has been established, the two leaf beetles, and um, they collect a bunch. They pot up some purple loosestrife. They put the beetles on the purple loosestrife, let them grow for a season, and then when the loosestrife is just covered in beetles, they wrap them all up in great big bags, carry these big pots out, and put them in a new wetland where there's loosestrife. And it's so low tech, and it's still really work heavy, but it is doable, and I really salute them for doing that. Um, but yeah, the emerald ash borer is very much all of our eggs are just in that kind of one basket in terms of the one lab that does it.
And it's tough because the biology of some of these insects. I mean, like a wood boring beetle is so much harder than, just a an herbivorous insect
Yeah,
because
yeah,
you need them to be wood boring inside in a lab, which is
yeah.
really difficult to replicate.
So you have to grow the ash trees, small ash trees, and you have to get the ash trees infested with the emerald ash borer, and then you have to introduce the parasitoids, and then you have to ship them out. And it's really it's really laborious and really expensive. Um, and even one of the simpler biocontrol agents, Suge, that is a private company on it's sort of a small company run by one woman, and she does an excellent job. But boy, she works really really hard, and the beetles are not cheap when you have to buy them, because it's so laborious to rear them. It's
Interesting.
much
Well,
easier to rear an insect that feeds on a plant than it is to rear an insect that feeds on another insect that you have to also rear that, which you have to grow on a plant. Um,
I can imagine. It sounds like there's franchise opportunities for anybody in Maine who wants to start some biocontrol rearing projects.
oh,
Um,
if you've got big money in your pockets.
um, so we're getting close to the end here. Does anybody have any last minute questions? Um, Either put them in the chat or unmute yourself. Um, if none, then we'll wrap it up for the night. Thank you so much once again, Colleen. That was a wonderful talk. Um, Lots of fun.
Well, thank you guys
And
for coming out
yeah,
on this.
and stay tuned, everybody, for our next webinar is going to be Thursday, March 5th. And that one's all about microscopes to get everybody ready for the bugging season, how to look for and maybe purchase a microscope without breaking the bank and getting something that is actually useful for you if whether you're a hobbyist or someone more serious about insect ID. So that'll be a really fun one. Thursday, March 5th at 7 p.m. Um, Once again, thank you very much, Colleen,
And thank you,
and
everyone.
everybody stay warm. Spring's on the way. Have a
Bye.
good night.